Vale.
Okay, can you hear me? Yes. So good morning, everybody. We're going to start with this thesis defense by Aitor Bañón González that has been supervised by Berta Alsina y Español with the title Cellular Dynamics and Molecular Mechanisms Underlying the 3D Organization and Connectivity of the Staticoustic Ganglion. So the members of the evaluation committee are, sorry if I don't, if I don't pronounce it correctly, Dr. Virginie Lecaudet from the Institute for Cell Biology and Neuroscience in the Goethe Institute, Vikas Trevendi from the EMBL in Barcelona, and myself, Eloisa Herrera, from the Instituto de Neurociencias de Alicante. So, as you know, you have 45 minutes and 1 hour to do your presentation, and then we will ask questions, and then everybody will go out. So I think that whenever you are ready, we're going to sit down now there because we can see. Yes. So go ahead.
Ahora lo hacen, ya lo he dicho. Sí. Hi, good morning, everyone. My name is Aitor Bañón González, and today I will be presenting my doctoral thesis, which has been supervised by Dr. Berta Alsina in the Developmental Neurobiology Lab. First of all, I would like to thank the members of my committee for accepting being part of my panel, and also all of you who decided to join me, either here or online, to hear about the wonders of cellular dynamics and molecular mechanisms on the organization and connectivity of the statoacoustic ganglion. The statoacoustic ganglion is this structure here, very complex, that it is formed by neurons. These neurons need to innervate, on one hand, two sensory hair cells of the inner ear and the corresponding nuclei in the central nervous system. The sensory hair cells are in charge of transducing mechanical information for the senses of hearing, as you are offering to me now, and balance, as I'm relying on now. These sensory hair cells are located in distinct sensory patches in this intricate 3D epithelial structure that composes the inner ear. This apparently whimsical structure is, in reality, fundamental to carry out the functions of the system. As you might have already imagined, since the statoacoustic ganglion is the relay station between sensation and perception, which is transmission, needs to also acquire the correct architecture to innervate the different and distinct topologically associated sensory domains. So then, a very fascinating question is, how such a complex tissue can be formed from a tissue as simple as an otic vesicle? An otic vesicle is a regionalized epithelium from which all the cell types that conform the inner ear will arise. In our model system, the zebrafish, the inner ear looks like this, quite similar to the one I just introduced you from mammals and humans, which are mammals. The statoacoustic ganglion, which lays behind of the inner ear structure, is composed of up to 3 different lobes. The neurons of the statoacoustic ganglion are also generated from an otic vesicle, from a called neurogenic domain, inside the vesicle. These neural progenitors, here in red, need to delaminate and undergo a series of cell behaviors in order to conform such a complexity. These behaviors are from delamination from the otic epithelium to migration of the cells, interactions between them, aggregation and coalescence to form the distinct lobes, and innervation to the different sensory patches. However, how this happens is practically a black box. That is why our question is to study the organization of ganglions, of gangliogenesis, in the paradigm of the otic vesicle, of the statoacoustic ganglion of the otic vesicle. So deciphering the cell behaviors that these neural precursors, which will give rise to the neurons of the sac, undergo and the molecular mechanisms that govern them is key to understand the developmental process of ganglions. Before we go through to introducing my experiments, I will give you a few tips so you can better understand the experiments that I'm going to show you. This is an otic vesicle. The neurogenic domain is here in red. In all my images, I will show you anterior to the left, meaning that the eye is on the left, and posterior, or the tail, to the right of the fish. Moreover, the otic vesicle it's going to be labeled by these white oval lines, either dashed or continuous, in all my images, okay? So, as I told you, these neural progenitors arise from the neurogenic domain. This neurogenic domain expands from more anterior regions of the otic vesicle to more posterior regions, generating a wave to posterior regions. At already these early stages where neural progenitors are being specified and delaminate, they start forming the primordia of the statoacoustic ganglion. At 24 hours, an anterior lobe can already be seen. And at 34 hours, a posterior lobe is present, meaning that in less than 2 days, you have the primordia of the statoacoustic ganglion lobes. Moreover, the tissue which is going to be central to my work are SAG, or statoacoustic ganglions, SAG, neuroblasts, or these neural progenitors, which are labeled by the expression of the transcription factor NeuroD, as you can see here. Okay, let's go back to the aims. So my aim is to study the organization of this ganglion, right? From all the topics that I have addressed in the thesis, I will only focus on these two, so to get you a better detail in this matter. So, we wanted to analyze the cellular migratory behaviors of this otic neuroblast in the formation of the anterior and the posterior lobe that I just showed you, and also the molecular mechanisms underlying this. However, in order to be able to address the first question, we need to address if these neuroblasts really engage into active migration. And you might think, but Aitor, if you are going to analyze the cellular migratory behaviors, these cells do migrate actively, right? It was not so clear. From literature, it has been assumed that these cells are like passive entities being pushed out of the epithelium, meaning that every movement that they undergo are more or less extrinsically carried out by pushing cells from neighboring tissues, like in a case like this. However, this idea might change soon. There are a group of molecules, of small molecules called ROGTPases, from which RAC1 is central in engaging the cells into active migration. So maybe we could tackle if the cells do undergo active migration by affecting this molecule that engages in the cytoskeletal machinery for active migration of these cells. How did we do that? To make a long story short, we introduced downstream the NeuroD, our gene of interest, because it is specified in our tissue. Under the regulatory region of NeuroD, we integrated a GAL4 molecule through CRISPR knock-in. So now GAL4 is going to be expressed in the tissue of the NeuroD gene. That generated the NeuroD GAL4 line. If we cross this line with a line that expresses downstream a USGS element, whichever gene, this gene is going to be activated by GAL4 in specifically this tissue, okay? So once that we have the transgenic line, maybe we can interfere with the cell machinery for migration using UAS dominant negative forms of these ROGTPases, specifically RAC1A. But before we needed to check that the expression pattern of this transgenic line recapitulated that one of the wild type. As you can see here, crossing our NeuroD GAL4 line with a UAS CAIDI, which is a fluorescent molecule, here in magenta. These are dorsal views of the zebrafish, and these are the otic vesicles, okay? If you compare the pattern of expression with the already very commonly used, and it is known that this line expresses the GFP in the endogenous regions of NeuroD expression, you can see how these patterns overlap, meaning that we can use this line for such purposes. Okay, so now I'm going to show you, trying to answer this question, if do SAG neuroblasts engage into active migration, I will show you a video in which you are going to see a wild-type cell, a control cell, migrating. Oi. Now. Okay? And now I'm going to show you a dominant negative RAC1 cell in the lower panels. As you can already see, it does not migrate that much. As you can also see summarized in these examples, in which in the first time point and in the last time point of tracked neuroblast, you can see by this white line the pattern of migration that they have followed. And comparatively with the control, in the dominant negative condition, RAC1 condition, these cells do not engage that much into active migration. When we put several tracks together, as you can see here, these are the controls. Comparatively, these cells migrate quite less. If we perform an analysis over effective distance migrated, which is a straight line between the initial point of the migration and the end point of the migration, irrespectively of their specific migratory pattern, you can see how there is a significant decrease. This leads me to my first result, which is that Rho GTPases are implicated in neuroblast migration, and thus neuroblast migration is an active process. So now we can go back to trying to analyze these cell behaviors in the wild-type condition, no? Now that we know that these cells migrate actively. In order to do that, we are using the transgenic line that expresses GFP in the endogenous tissue, so to know the behaviors of these cells. But one thing that you might realize already is that the sac becomes too highly packed and dense to see single-cell behaviors properly. So in order to solve this question, what we do is using the Crispr system. At one cell stage embryos of this line, we injected a Cas9 with a guide RNA targeting the GFP. It would disrupt GFP expression, and just a few cells will remain labeled. So to analyze the shape changes and the migratory behaviors of these neuroblasts. This methodology was called Crispr eraser. Under Crispr eraser conditions, we were able to track several neuroblasts from all the neurogenic region, either more lateral delaminating neuroblasts under tracks, as you can see here, and also from more lower or ventral domains of the neurogenic domain. These are just two examples, but if we put all the tracks together, you can already see how there is like a tendency. Here you have all the tracks, here's just some of them from the lateral domain, and here's the medial tracks, some of them from the medial domain. In the polar plot, you can more clearly see that the delaminating neuroblast migration is directed. And you might ask now, towards where? When we track several of these neuroblasts in the same embryo, we realize that irrespectively of their origin inside the neurogenic domain, they tended to aggregate towards a region, here shown by an asterisk, that we called the Coalescence region, which allowed the formation of the anterior lobe of the Staticoustic ganglion. Moreover, we saw that some of these cells can escape the traction force of this region and escape towards the anterior lateral line system. The anterior lateral line system is a system only present in teleost fish and is devoted to sensing water movements. And it develops in close proximity to the ganglion. So now I'm going to show you the videos of these tracks. If you see only the upper panel, you are going to see how they tend to aggregate around this region, except the one that is going towards the anterior lateral line. And if you see now the lower panels, you are going to see the real cells. If you fix your attention on the white arrows, in which you are going to be able to see, choop, choop, choop. How are they going there? Meaning that the laminating neuroblast coalesce anteriorly to form the anterior lobe of the Staticoustic ganglion. But if all the neuroblasts come from the delamination from the neurogenic domain, what are already doing these cells here? What are these cells? Are they important for the organization of the lobe, since all the cells tend to aggregate around here? In order to address which population was this from, we performed in vivo experiments at very early stages of development, even before any delamination event can start from the otic vesicle, which in earlier stages is called the otic placode. At these early stages, we saw that anteriorly to the otic placode and posteriorly to the trigeminal ganglion, some cells were expressed in a scattered pattern that would migrate to anterior regions of the aortic vesicle and initially populate the coalescence region. So as a result, the coalescence region is populated by these first or pioneer SAC neurons originated outside the neuroepithelia instead of inside as neurodepositive cells at early stages. These cells were already known to exist, but thought to contribute only to the anterior lateral line system. And now we are showing for the first time that these cells also contribute to the formation of the ganglion. So the next question, you might wonder, okay, Aitor, but if the delaminating neuroblasts go to the coalescence region and these cells do populate the coalescence region, maybe they are important for organization of the SAC. In order to address this question, we generated a transgenic line in which, again, in our tissue of interest, NeuroD, we express a photoconvertible molecule, fluorescent photoconvertible molecule called kikume. At early stages of development, where these pioneer neurons reach the anterior region of the aortic vesicle, these cells were photoconverted and then photoablated, meaning that they were killed. The photoconversion was just a check that at later stages that these cells were really killed because of the absence of the magenta signal at later stages would mean that these cells have been properly killed. After killing these cells, we would be able to, at later stages of development, see the development of the statocoustic ganglion devoid of these cells. So now I'm going to show you the first result. You are going to see 3 different conditions by column and different time points depending on the row from these conditions. In the no-ablated condition, at 16 hours post-fertilization, when the pioneer cells reach the anterior aortic vesicle, there is no change because they weren't ablated. But in the partial condition, in the partial ablation condition, some of them were killed, leaving some alive. And in the total ablation condition, they were all killed. Surprisingly, at 24 hours post-fertilization, we can see a lot of changes. Comparatively to the control, you can see that not only a reduced number of cells seem to be populating the statocoustic ganglion, but also the shape is aberrant. Not only that, the presence of this first or pioneer axon is absent in both conditions. At 34 hours post-fertilization, when the posterior lobe should have been formed, comparatively with the control, you can see how it is absent in both conditions. This leads me to the result that ablation of pioneer SAC neurons affect SAC development somehow, in many ways. In which ways? Quantitatively, as you might have guessed already, the number of cells populating the statocoustic ganglion is reduced, and that is why also the volume is reduced. The presence of these axons is also reduced, and the migratory properties of the neuroblast is altered. As you can see, comparatively with the control, the dispersion in the x-axis of these migratory cells is lost, partially at least. And the directionality that these cells should follow is kind of wrong, meaning that ablation of pioneer SAC neurons affects SAC shape through neuroblast migration, cell number, and axonogenesis. The data from these experiments come from time-lapse videos like the following one that I'm showing you, in which from both the lateral and medial neurogenic domain, comparatively you can see a control cell migrating. If you follow the yellow line, it tends to go to the coalescence region, whereas in the ablated condition, it does not migrate that much. Moreover, in this example, in which the medial cell also tends to go towards that region, in the ablated condition, they go to aberrant positions, even at the contrary. These results on ablation of pioneer SAC neurons affect neuroblast aggregation and consequently the anterior lobe formation. Moreover, if you remember, I told you that under ablation of these pioneer SAC neurons, the pioneer or first axon was lost. As you can see comparatively, the wild type, and here you don't see the axon. At later stages of development, where the posterior lobe should be formed, it is misshaped or even completely absent. We then thought that these two phenomena could be linked. Why is that so? Because if you see in this time lapse, in this vertical time lapse, the cells that populate the posterior lobe, here pseudo-colored in magenta, take over the regions from where the axon is located. If you take a spatial section of the axon and put it across time, you can see how cells start populating these posterior regions. Moreover, if you put your attention here, you will see in this orthogonal view the relationship between the axon and the cell being very tight. And also in this view that I'm going to show you, you are going to see how the cells crawl on the axon, performing axonophilic migration to form the posterior lobe. And maybe I can play it again because I really like this video. So the laminating neuroblasts at later stages after coalescing, they can crawl on pioneer axons to presumably form the posterior lobe of the sac. So is there a way in which we can address the formation of the posterior lobe without killing the pioneer sac cells because they are required for the coalescence of the anterior lobe? Is there a way in which we can affect the pioneer axon extension without affecting the pioneer sac neuron's presence to observe only the effect of the pioneer axon on posterior lobe formation? We started thinking towards which cells do neurons of the statoacoustic ganglion project? Towards hair cells, right? So maybe if we disrupt or ablate the specification of these hair cells, we could get to this hypothetical condition in which we could have the cell, but not the axon. But don't go so fast. First, we have to check several things that we have assumed. Do these pioneer sac neurons produce the pioneer axon, or they are helping other cells to do that? And do these axons really target hair cells? In order to answer this question, we used again our photoconvertible transgenic line. We photoconverted the pioneer neurons at early stages, and guess what? At later stages of development, if these neurons produce the pioneer axon, it should be magenta, exclusively magenta. Not only that, if you cross the transgenic line neurodicicumi with a reporter of hair cells, A21 GFP, you can already see here and in the orthogonal views the close relationship that exists between the axon and the hair cell. And also in this video that I'm going to show you, in which the axon will grow towards the hair cell and anchor to it in its movement towards more anterior regions. See? So indeed, the pioneer sac neurons produce the pioneer axons towards hair cells. Okay. So then, can we disrupt hair cell specification somehow so that we don't have the pioneer axon and the posterior lobe formation? In order to do that, again, with our neurodicicumi line, crossing it with a reporter of hair cells, brain 3C, as you can see here, this is the posterior region. In a zoomed-in image, you can see the hair cells tethered to the otolith, and here, the ablation, the precise ablation of only these cells with a multiphoton. So here you have the control. We photoconverted the cells. At the later stages, the axon very beautifully progresses towards posterior positions. What could happen in the ablation of the hair cells? So the axon still grows. Well, not only that, when we ablate in ventral regions from where this axon needs to go through towards posterior positions, even though we assume that it can no longer sense any attractant from here, the axon still grows. This for us meant two things. One, hair cells are not required for pioneer axon initial outgrowth. And two, pioneer axon initial outgrowth might be independent of any external signal, or at least from posterior regions. Okay, this is a fine result. But the bad thing is that we are back to the initial point, in which we cannot rely on this situation, neither we get the lack of the axon when we ablate the pioneer cells, the hair cells, sorry. So, again, is there a way that we could get this situation? We started looking at the bibliography, and we found several interesting molecules. I'm going to show you two. The first one is CXCL14. It's an orphan chemokine playing a role non-cell autonomously upon axons and migrating cells. To our surprise, when we check the expression pattern of this molecule, as you can see here, it is not only expressed in hair cells of the anterior and the posterior macula, but in neighboring regions of these hair cells, meaning that if CXCL14 is the molecule exerting this role, in the previously condition where we ablated only the hair cells, we won't be downregulating completely the tissue that expresses CXCL14. Not only that, to go even further, we could see that it is specifically expressed in the posterior hindbrain entry points and in the anterior hindbrain entry points of these axons. As you can see in this full image, the hindbrain is labeled in this white line. These are the entry points. And these are the hair cells, the anterior and the posterior. So CXCL14 is expressed super specifically in axon target domains, including peripheral and central target regions. As you can also see here, if we see the expression pattern of NeuroD and CXCL14, you realize in the insets how all the axon targeting tissues are CXCL14 positive. So, could be CXCL14 implicated in pioneer axon extension? If so, does the lack of CXCL14 affect indirectly the formation of the posterior lobe? And now I have to stop 1 second. Una más. Okay, so how are we going to analyze the lack of function of this molecule in producing the axon and the affectation on the posterior lobe? In order to get fast and reliable results, we resorted on using Crispr again. In this case, at one cell stage embryos, we injected a Cas9 with a guide RNA targeting the locus of, in this case, CXCL14. In other cases, any gene that you want. This would prevent the correct translation of the protein and the degradation of the mRNA. When checking by detection of CXCL14 mRNA, as you can see, comparatively with the control, in the knockout, we have virtually downregulated all CXCL14 expression. So, going back to the question, is CXCL14 needed for pioneer axogenesis? I'm going to show you 2 control videos, but just pay attention to one of them. Maybe we can say the left one. Okay. The axon in the white arrowhead grows to posterior. It is defasciculated here, like doing 2 branches, but it's the same axon. Now I can play it again. You can see the other video. And even though the axons grow a little bit differently, they are quite consistent, right? Reaching these positions. However, if I show you the crispans, if I show you the crispans, the axon, it is not properly produced, and it also retracts and keeps even more the fasciculation. If I play it again, you can see the other video. They do not reach posterior positions, they even retract. So, when we measure several properties that we associated to a correct pioneer axon extension, you can see comparatively in the control and the CXCL14, either in the axon presence, in the correct targeting, in the axon persistence, and in the correct shape, that all of them are affected in the CXCL14 F0 knockout or the Crispan. Except somehow the axon presence, which we still think is independent of any extrinsic signal, which makes sense. Moreover, for the axons that make it to posterior positions, comparatively with the control, the axons in the Crispan condition take longer. This results in that Crispans cause impaired axonogenesis. Okay, so we don't have the axon. Nice. Do we have the posterior lobe? Yes or no? As you can already see here, this is the statoacoustic ganglion. This is the anterior lobe. This is the posterior lobe. What happens in the Crispan? Not so much present, right? I'm going to show you now a video associated to these images in which, in the upper case, you are going to see the wild type. The posterior lobe will form inside the white box, and in the CXCL14 condition, knockout condition will be formed around here. As you can see, several cells start populating the region here. Here not so much, or not very consistently along the direction of the axon should be. This took us to the conclusion that CXCL14 F0 knockout causes disrupted posterior lobe formation. But we were saying, okay, we want to go further. Is there a way in which we can tackle the endogenous machinery for these axons to grow, and we can push further the phenotype? In order to do that, we found this molecule, contactin 2, sorry, which is a cell adhesion molecule known to play roles, I can do it with this, axon growth, axon guidance, and also axonophilic crawling, which is our case. It is expressed in the statoacoustic ganglion, as you can see here in this asterisk. Here I'm not showing the optic vesicle, but it should be like this. Okay? So, our working model is either the axon grows and the lack of contacting homo or heterotypic contacts makes the axon not crawling, or in another situation, the axon growth is still affected, the axon does not grow, and the posterior lobe is not formed, in which case we could say that contactin 2 is important for these processes, but not through direct axon, not being the precise molecule we don't know for the axonophilic migration. So trying to answer the question, is contactin 2 needed for pioneer axogenesis? As you can see here in the control, the axon grows. In the Crispr, it doesn't grow. When we compare the same properties that we did in the CXCL14 condition, all of them, by 3 independent experiments, all of them are affected. Even more the axon presence, meaning that indeed the intrinsic machinery that is taking part there is more affected. But still, the axon forms in many of the cases. And as I'm going to show you here in the video, reproducing the one that I showed you before, oh no. Just pay attention to one of them, maybe this one. Again, the axon grows. The other one practically does the same, very consistently. And again, this is already the result, the final result, because the video is in the end. But you can see how the axon is not properly growing. It is also retracting, not reaching to posterior positions, and also at the central level, it is a complete mess. So, well, pioneer axon extension relies also on contactin 2. Does this affect also, or do we get a further phenotype in posterior lobe formation? As you can see here, even though there are still some variabilities in the controls, in the contactin 2 F0 knockout, the formation of the posterior lobe is completely dysregulated. As I'm going to show you in these very beautiful videos, I like them very much, you are going to see this is the nuclei staining of this sac neuroblast. You are going to be able to see here the formation of the posterior lobe. And in this context, in which we have used our photoconvertible transgenic line, you can see the presence of the axon and how cells from here take over the region of the axon. Just see one of them, and then I will replay it. Or if you want to see both, but. So as you can see, cells start populating this region. The axon is there. And the cells start taking over, right? Now, if I show you the Crispr, already you can see how the pioneer is not there, and also the formation of the posterior lobe is disrupted. Some cells very shallowly migrate. Okay, when we measure several of the properties associated to the posterior lobe formation, for instance, the number of cells populating the posterior lobe, there is a significant decrease, and the hours that these cells take to invade posterior regions is increased in the contactin 2. Meaning that posterior lobe formation is also compromised in contactin 2 F0 knockouts, pushing the phenotype that we had in CXCL14 condition. Sorry. So, recapitulating the whole story. Until now, I have shown you for the first time that extraotic pioneer sac neurons are the first neurons populating the sac, from which the most relevant conclusion, these are the pioneer sac neurons, from which the most relevant conclusions is that pioneer sac neurons and their axons act as scaffolds for sac lobe organization. To this matter, the laminating neuroblast coalesce to form the anterior lobe and perform axonophilic crawling to form the posterior lobe. They do it in a directional migratory manner, irrespectively of their origin inside the neurogenic domain, and in a raw GTPases-dependent migration, an active migration that it is also shown by the first time. These are not passive entities. These neuroblasts really go where and when they are needed in the formation of the different lobes. So a lot of questions remain, always a lot. A lot of questions remain open. For instance, which is the molecule that attracts the pioneer neurons towards their position? Or which is the attractant from pioneer neurons to the rest of the delaminating cells? In the end, they all form part of the same tissue. Is the molecular signature of these pioneer cells different from the delaminating neuroblasts? Or it is just a matter of time specification? What drives the change in behavior between anterior coalescence and posterior axonophilic rolling? But well, and also, which are the molecules implicated in these processes? What it is clear is that if we disrupt the presence of the pioneer cells and their axons, the formation of these context-dependent behaviors can no longer happen correctly. Comparatively with our system, what we do know from literature is that pioneer neurons have long been known, from seminal studies done in Drosophila and then further studies in which they saw that pioneer neurons are not only required for scaffolding of later forming axons, but also for the positioning of later forming neurons. In the zebrafish, a group of pioneer faciobranchial motor neurons are required for later forming faciobranchial motor neurons to reach their correct region. If you see here, in the control, these could migrate well. In the ablated condition, as I did, these cells do not migrate that well. There are also studies done in rat and mice. However, the in vivo properties are kind of lost. But you can also see how some neurons utilize trigeminal axons to go from the dorsal midbrain into the hindbrain, and also there exists the model of the gonadotropin-releasing hormones neurons that need to migrate long distances from the olfactory bulb to the hypothalamus over axons. So all these... me he perdido. So all these phenomena, sorry, I wanted that word. All these phenomena could be modularly used in the different systems and by different neuronal tissues in order to provide correct neuronal segregation or aggregation and targeting. Also from literature, the molecular tools implicated range from chemokines, adhesion molecules, and also other tissues like, for instance, neural crest cells. To this matter, we have analyzed the novel role of two molecules, one being the chemokine CXCL14 and the cell adhesion molecule contactin 2, which are important for axonogenesis, and posterior lobe formation in a non-cell autonomous manner and cell autonomous manner, respectively. A still open question, but that could be very interesting to address, is if there exists some parallelisms and differences of these molecular mechanisms in other systems. In summary, my contribution to the field with my work has been devoted to generate techniques at the service and for in vivo analysis, in this case of our paradigmatic tissue, ranging or with the basis of high spatiotemporal resolution confocal imaging, Crispr, photoconversion, and photoablation that allowed us to analyze the cellular behaviors the SAC neuroblasts have, the molecular toolkit, part of it, in which these cells rely on to perform context-dependent responses such as coalescence towards pioneer neurons or axonophilic crawling. In the end, my work provides new insight into knowing the mechanisms of morphogenesis of neuronal tissues to attain their final architecture. And now, with the permission of the committee, I will read through the conclusions. So, the SAC is composed by delaminated otic neuroblasts and pioneer SAC neurons. pioneer sac neurons originate in the posterior placoidal area and migrate to anterior positions of the otic placode. The laminating intraotic neuroblasts coalesce around pioneer sac neurons at early stages to form the anterior lobe of the sac, while later the laminating intraotic neuroblasts crawl on axons to posterior regions to form the posterior lobe of the sac. The delamination process of neuroblasts follows similar steps to the ones described in other systems, including an apical abscission and polarity protein recovery by the cell body, resembling an EMT-like process. Some of the conclusions I didn't go through in my presentation, but they are there in the thesis, like this one. Neuroblasts produce membrane changes such as blebs, lamellipodia, and directed philopodia during directed migration. The laminated neuroblasts engage into non-collective Rho-GTPases-dependent active migration. Pioneer sac neurons and pioneer axons are required to organize the sac. Pioneer sac neurons extend and anchor the pioneer axons to hair cells of the posterior and anterior macula at around 22-24 hours post-fertilization. But pioneer axons extend independently of the presence of hair cells, neurotrophins, and FGF signaling. Neurotrophins are important trophic factors for sac neural survival and correct axon anchoring, but dispensable for axon initiation. CRISPR-Cas9 F0 knockout is a reliable, rapid, and consistent technique to analyze the lack of function of molecules without compensatory effects and little mosaicism. CXCL14 is a key molecule regulating in non-cell autonomous manner pioneer sac neurons migration, neuroblast migration, and pioneer axonogenesis. And last but not least, contactin 2 is an important molecule regulating pioneer axon growth cell autonomously and sac posterior lobe formation. And with that, I would like to thank all the people from Berta's lab, the present and former members. I would also like to thank people from Christina's group, the technicians, the people from the different facilities, the funding, my family and friends, and all of you. And now, I will be happy to take questions.
Thank you, Aitor. Thank you very much. Very nice. So we're going to start with the questions. I think that Virginie is going to start first. Go ahead.
Okay. So thanks a lot, Aitor, for this very, very nice presentation. It was a really, really good talk, really clear. You managed to really summarize nicely this very big thesis that you wrote.
But it is only until page 186.
Yes.
The rest is also only supplementary.
Okay, so let me see how I actually start. So maybe about the origin of the pioneer neuroblast. So as you just re-mentioned here in your summary, you have those cells that come from this placoidal region where actually your entire system comes from originally, right?
Yeah.
So what do you think makes a difference between the cells that will basically stay there, become this pioneer, and the rest of the cells that will actually form the placode and all the rest, right? Do you have any idea why you have basically a small cluster which is staying, not integrated into the rest, and has actually a very, very important role for the entire formation afterwards?
Okay, so I think that the preplacoidal region is all a field with the capacity to generate neurons. So maybe if it were only the neurons from the placode, the ones being specified, maybe it would already been too late for the proper formation of the ganglion. Like maybe the axon needs to be formed at that point, and only cells specified before are able to do it. So that is why somehow the system integrated through evolution, making that these cells that could go anywhere to the anterior lateral line or whatsoever, they said, no, we need them here in order to engage the rest of the cells into proper formation.
So that
because they can interchange also between the anterior lateral line and the ganglion, but somehow these cells are needed there. So it doesn't matter if it is the cell here or the cell here, but just one that it's being specified there that performs the function.
And so
I don't know if that answers your question.
Maybe I just go on and then maybe it's becoming more clear. So those cells are neurodepositive before the first delamination process, right?
Yeah.
And the cells that delaminate from the aortic placoid, they only become neurodepositive when they start to delaminate.
Exactly.
Okay, so this is really like cells that somehow have acquired their identity early on. The pioneer. Is that, yeah?
Yeah.
But and then in this case, if we look, they're different to the anterior lateral line ganglion, future ganglion. Here, what is the difference? Because they are also geographically very close to each other, right? They seem to have very similar properties because they seem to also be able to attract the neuroblasts that are delaminating. Do you know what makes the difference?
I think the difference comes later. Like, you just need some neurons to be there. Some would go to the anterior lateral line primordia and some to be the pioneers of the SAG, but this specification would come later. I mean, maybe that they are neuro-D positive is just this modular thing that tells them, okay, you are going to become a neuron, but not specifically from any tissue until later.
Okay. Then I had a question. When you ablated the pioneer neuroblast, because in the normal physiological condition, you have some errors, so some neuroblasts that will go to the anterior lateral line. When you ablate the pioneer, are much more neuroblasts going to the anterior lateral line?
We didn't check that, but we have some results at later stages in which you can see how the statocoustic ganglion kind of recovers, and my idea is that some cells specified in the anterior lateral line sense in this preplacoidal area or whatever that there are still some cells lacking, and maybe they proliferate more, and they will try to compensate the lack of these cells. Also, the ablation protocol sometimes can be a little bit tricky, so maybe in one case I never ablate like hundreds of cells. Normally, I have checked that it is just 5, but maybe sometimes could be 8 or 9. And maybe these cases are the one that, for some reason or time point, like we could recover this if it was only a lack of 3 cells and not 7 cells, and at this developmental stage and no further on. So maybe if I ablated a little bit later in time or a little bit more cells, this compensation cannot happen, but we didn't check that.
So, but this compensation, so because you, maybe you already answered this question and I did not understand, but so one of the main phenotypes you have after the ablation is that you have a much smaller ganglion. And this cannot be explained by the few cells that you ablated.
Exactly,
right?
yeah.
So what do you think is happening here?
Okay, sorry. So it has already been described from this population also of extraotic pioneers. There is a small subpopulation that are positive for neurogenic, which is a transcription factor that it is expressed before neuro-D. They ingress into the aortic epithelia and they promote a neuroblast specification there. So if these cells being neuro-D, meaning that they are more specified and by that they cannot ingress maybe, but they could still have this function of a specification of further neuroblasts inside the aortic vesicle, and without them, this cannot progress.
Okay, so they're really, there's much less delamination, basically, when you...
Yeah, we didn't check. I should have checked the number of cells that remain inside the aortic vesicle, but maybe it's very tricky to address because maybe if they don't get specified and you get a number of cells inside the aortic vesicle, they also send signals for not proliferating, so maybe you won't have more cells, even though they are not delaminating. But it could be an approach.
But you can count the number of delamination events, or you can...
Yeah, I could do, you're right.
So at the moment you cannot separate between, it could be a problem of proliferation within the statoacoustic ganglion?
Sorry, can you repeat the question?
You could, so there's several hypotheses, let's say, to explain that you have a much smaller statoacoustic ganglion. There could be a problem of proliferation, a problem of not enough cells delaminating, or I don't know. So you don't know at the moment.
I don't know.
Okay.
I think that somehow it could come like in a pack. Like they get specified, they can further divide, so it could be a problem of proliferation and also lack of delamination, but we didn't check.
Okay.
The only thing that I checked is that inside the neuroepithelia, there are no more neuroblasts there. Like, it is not a thing that they do not delaminate once they are neuro-D.
Okay.
But that is the only thing that I checked. They do not remain inside because maybe, no, if they remained inside, they cannot migrate properly. And that is why I get this mid-shaped sac. But they are not more neuro-D positive cells inside the...
Okay.
But again...
I had also a question about this. Probably it was described before. This was absolutely not the center of your work, but these ingressing cells back into the otic vesicle, I find it really fascinating.
Yeah.
So what is it? Is it some kind of MET? Or so how can those cells repenetrate an epithelium?
Yeah, I think that these cells, they enter when the epithelialization of the otic vesicle is not complete.
Okay.
So maybe it's kind of an interface. These cells are there and they send signals that tell them, okay, I have to become epithelium, but in the end I will end up delaminating because I am neurogenic and I'm supposed to be a neuron. But it happens in such a time window that they can still do that.
Okay, but it could be that actually those cells that are ingressing, they could be the key actually. Not so much this pioneer.
Yeah.
But these cells that are ingressing, if they don't do that, they block the whole system.
In my ablation protocols, it could be that I have killed some of these cells and the effect of the lack of the neurons is due to the lack of these ingressing cells.
Yeah. Okay, so this process of the pioneer, this ingression, it really
can I say something?
Yeah, sure.
But I performed several experiments in which I saw the presence of neuro-D and neurogenic. Like normally the ingressing cells are exclusively neurogenic. And the ones that are not ingressing are, irrespectively if they can co-express neuro-D, it can be neurogenic and neuro-D or only neuro-D. So when I ablated the cells, I just went for the neuro-D cells, tried to.
Okay.
So at least at the physical level, I tried to be away from the neurogenic ones.
So then the ingression should be normal?
Should be normal. Okay. But the ablation protocol somehow is a little bit, so I cannot know if the heat heated the neighboring cell somehow. Yeah. So maybe.
And so you have no idea at the moment of what could be this. I mean, it's probably a chemoattractant that the pioneer cells are secreting. Do you have any candidate in mind of what this could be?
No, I think it could be several things. It could be a chemoattractant and or also homo or heterotypic contacts with cell adhesion molecules, because we have seen, although only qualitatively, that these neurons project to the hair cells, but also extend like philopodia-like structures, because in the otic vesicle, the hair cells are kind of here, the pioneers are here, they should project like this. Yeah. But they also project towards like lower regions. Yeah. So maybe some kind of homotypic connections so that the aggregation of the tissue is also proper. There is this work in which they show that ETG-A5 alpha, I think, like an integrin, makes the epibranchial placode like go aggregating, but not neighboring placodes. So maybe a specific molecule, adhesion molecule in the tissue could be contacting to some extent, or a chemoattractant, but I have no idea. I wanted to check the chemoattractant hypothesis, like very roughly, like trying to inhibit the well, I didn't do that, but it was only an idea, to try to inhibit the exocytosis machinery with RAP5 dominant negative things, and like if they are not able to secrete, but of the candidates, I don't know.
I'm also asking that because the process made me, or reminded me of, what happened in the lateral line. migration at the very beginning. So it has been described that there are also some cells that are in front of the lateral line primordium, which probably play a role in the initiation of migration and then go back into the primordium and then migrate with the group. And I think there's really some similarities between this and your system.
Well, indeed, I was so silly because I was not thinking about it, but in the CXCL14 condition, in the presentation, I only showed the thing of the axonogenesis and the formation of the posterior lobe. But I also performed an experiment in which I tried to address the migration of the pioneer neurons upon the Crispr condition. And they kind of migrate incorrectly. My guess is that, well, but this is going, I mean, it's going very far away, but CXCL14 could be a modulator of the CXCR4, the DSDF1 pathway, only for these pioneer neurons, because afterwards, in the delaminating neuroblast, these cells do not express the components of the pathway. But at least for the pioneer neurons, to reach the anterior positions, are maybe...
But in your CXL14 Crispr, did you see a strong phenotype in the formation of the anterior lobe of the staticoustic ganglion?
A strong phenotype? No.
Okay. That's mainly axogenesis, the phenotype of the CXL14.
Axogenesis, yeah, and the lack of the posterior lobe.
Yeah.
It is also true that, but I didn't go to such detail because I was focusing only on the posterior lobe and the axon thing, but the anterior lobe maybe could be also a little bit misshaped regarding the CXCL non-count.
So, I mean, my question actually was, do you think that FGF could play a role here? Because in the lateral line, it's actually FGF that is expressed in those cells that are in front.
I performed this experiment. Okay. SU5420, like this FGF inhibitor. Yeah. And I also used that experiment as a control for the Crispr condition regarding the pioneer neurons migration. And in the CXCL14 Crispr, I saw this mismigration, but in the FGF abrogation, I didn't see such effect. Like cells would migrate normally. Okay. So in principle, through this drug inhibition, FGF is not implicated.
Okay. Maybe about then, if we are on CXL14, I was wondering also if you tried. So you tried to also overexpress CXL14 to figure out how it's working, but you mainly overexpress it in region where you think it's endogenously expressed. So the hindbrain and the statoacoustic ganglion. But did you? I was thinking, did you try to overexpress it, for example, in the trigeminal ganglion?
Yeah.
Where you would completely deroot, and this would maybe give a clearer answer.
Yeah, no, I didn't try because we didn't have at that moment the transgenic line. Or, well, in reality, the neurodigal 4, it is expressed also in the trigeminal ganglia, but since it is not only exclusively from the trigeminal, so we didn't have that line. Yeah. So I could just use the neurodigal 4 for the overexpression in the ganglion and the CROX20 GAL4 for the overexpression in the hindbrain. Yeah. But in the hindbrain, I think CXL14 is not expressed. It's more of expression in epithelial kind of tissues, like.
But it's expressed in the entry point.
In the entry points, yes,
right.
yes.
So,
but I overexpress it in the neurons inside the hindbrain.
Yeah. I mean, I think you have quite some argument to think that CXL14 is acting as some kind of chemoattractant, but the best would be to try to, I think, express it in a completely ectopic, I mean, tissue not too far away, of course.
We were thinking also in complicated ways, like, I don't know, beads coated with CXL14, putting them in front of the ganglion and see if they deroot.
Yeah, and this do that maybe in a background where, in a CXL14 mutant, actually, you know, to remove the endogenous source, and then...
We are having this mutant. It's 3 months old, so we are going to be able to use it, but we still don't have homozygotes, but yeah. Yeah.
And then maybe last question, and then we can keep the... for the CXL14, so in your introduction you mentioned, and I kind of forgot the detail, but that there has been studies showing that, so it's an orphan receptor, but it has been shown to somehow interact with the CXL4, CXL12 pathway. Can you maybe remind us briefly what has been shown and how this could maybe play a role in your system?
Yeah. So the thing is that in the few studies that address the role of CXCL14 regarding the CXCL12 and CXCL4 pathway, is that they are competing with the receptor, and thus it's like an inhibitor of the pathway.
Yeah.
So that is why also it was difficult for me, even though we have a strong phenotype in which the lack of CXCL14 has some effects in which you can think that it is an attractant. I tend to think that it is rather an inhibitor of something else that doesn't allow for instance, the axons to grow in my case. So yeah, the thing is that what they have shown is that they compete with.
So it
with
can bind to CXCR4.
Exactly.
Okay, and where is CXCR4 expressed in your system?
In the pioneers. Well, we don't know because we didn't check, but there is a paper in which they say that it is expressed in the posterior placodal area from the anterior lateral line and everything. And since we described the pioneer neurons that are already there, it might be possible that they are expressing CXCR4, but we didn't check.
Okay. Okay. Yeah, maybe.
Okay, thank you, Brigitte. So now, Vikas, go ahead.
So first of all, congratulations to Aitor for such a nice thesis, and to Berta as well for this. The presentation was very clear.
Thanks.
I would like to make one specific comment, which is I really like that in the beginning you guided your audience with the fact that okay, in all my following slides there will be an oval which is either dotted. It's a nice practice, especially for an audience that could be heterogeneous. Okay, so many of my questions were already asked, but I have a list of other questions. So the first thing is that in your system, in the aortic vesicle, it is an oval-shaped thing with an anterior and a posterior side. So by default, what are the, let's say, expression of genes, or what decides the anterior and posterior in it, in the absence of anything else?
Yeah, so it is. I have also seen, and I think it is described that up to some point it has no anterior or posterior identity, and then also the neurogenic domain. Some of the neurogenic positive cells can be expressed in posterior regions, even though they
exactly.
in the end become different things. So I think it has to be something extrinsic, like FGF or retinoic acid, like.
So so you tell me maybe, but to me it seems that based on my limited understanding that the anterior and posterior specification is very concomitant with your is along the same time, that the timeline that you are studying. So that is when the organ itself acquires an anterior posterior polarity. So how is it? So in the first place, when you have these pioneer cells which are making contact on the anterior side, what gives them the directionality in the first place?
Yeah. So I think there are several things here. Like the aortic vesicle, I don't know if it exactly acquires the anterior posterior identity at exactly the time, because maybe these pioneer neurons are migrating to what will be the anterior position, but maybe it is not yet the anterior position. Well, it is becoming, because actually the posterior part is already epithelialized, but the anterior it is not, and this is thanks to FGF and retinoic acid signaling. So I think that it might be a different tissue, like for instance, I don't know, the hindbrain, or something helping that. If the aortic vesicle has not acquired yet the specification needed for the pioneer neurons, it should be something else. Like for instance, I don't know, a signal from the hindbrain, or the mesenchymal tissues, or I don't know.
I see. I mean, the other thing which I felt, and maybe you tried to look at it and it did not make it into the thesis necessarily, is that so the posterior side, it starts epithelializing, correct? So as it is epithelializing, it will have a different let's say properties in terms of interstitial space, etc., as compared to the anterior side, and that could lead to accumulation of morphogens or things like that. Did you have a chance to look into the epithelialization simultaneously with all these processes that you described in your thesis?
No, I didn't look. I mean, I just looked in which was described that the posterior part was already epithelialized in the moment when some of the pioneer neuroblasts, neurons ingress, but no, I didn't.
Okay. Then going back to the very beginning of your talk, so you talked about the SAG neuroblasts, they do active migration. So what is the alternative? Because the movies that you have are so spectacular that I see it and I feel that this is active migration. So what could be the alternative?
The alternative?
If not active migration.
Well, in the end, it could be like this neuroblast, you have a specification from anterior to posterior of the neurogenic domain. So anterior delaminating cells would stay there, posterior delaminating cells would stay there, and then form the anterior ones the anterior lobe, the posterior ones the posterior lobe, and then it would be just a matter of producing axonogenesis. And then when establishing the connection with the central nervous system, acquiring like the functional importance, like this neuron is going to be vestibular, this cell is going to be auditory. But I think proving that it is active migration gives more robustness to the system. Like if there are not enough cells at some point, it could be rescued by the migration of these cells because the whole population will kind of compensate itself. If it was not active, maybe, you have some cells here, the neurogenic domain in the posterior part does not generate properly, there are not enough cells there, and maybe the anterior ones cannot target the posterior regions or don't have the scaffolds to reach there or something like that.
Okay. Then, good. Also, what decides actually, so you have these axons which are growing, and you show that in the absence of contactin 2 or CXCL14, they don't grow. But the fact that if they grow axons and they have to reach to the posterior side, what is the cue there?
So what I think is that these pioneer neurons, once they are specified as you are going to become a neuron independently of the cell type, you want to do axons. So the thing is that there are some repellents from the dorsal part of the osteotic vesicle, like SEMA3, and I think also from the neighboring regions, there are some repellents for this cell. So the only chance, they do not necessarily know where to go, but they know where to not go. So they start producing the axon, and then in their way, they find neurotrophins, for instance, in the thesis, neurotrophin 3, BDNF, NGFA. So this keeps facilitating the growth, up to the point that when the axon growth is no longer intrinsically regulated, it says, okay, I have, so the thing would be like, the axon starts growing, then finds neurotrophin, I can follow. And then it attaches to the hair cells thanks to neurotrophins, and also this growth that is not dependent, not intrinsically controlled, could be also due to CXCL14.
I see. And then, so if I understand correctly, so what you are saying is that CXCL14 and contactin 2, they are basically responsible for the axons to be able to, or the cell to be able to make axons. But where it goes, that is completely determined by its surrounding, or the signal that is coming extrinsically.
Yeah, I think that also provides robustness to the tissue. Like if the hair cell was generated somewhere different, then the axon would be able to like deroot a little bit, and this would provide also the scaffold for the migrating neurons, and say in the end you will have the whole shape correctly.
Okay, so since you use the word scaffold, that brings me to my next question. So actually related to also what Ginny asked. So when you say scaffolding, molecularly what do you mean? What is happening there?
So what I think is that the axon provides the structure for the neurons to know, okay, I can go through the axon, through maybe homotypic or heterotypic interactions with contactin 2 or other molecules. But there is one thing, that the directionality, how it is provided, that thing I don't know.
So but so you have one axon, okay, and you have many cells which are in contact with it. So help me understand this.
Okay.
So you showed that there are many cells which are migrating to the posterior lobe. So how is this achieved so robustly? Because for that to happen, even if there are homotypic contacts, meaning the contacts have to be made, broken again, so that all the cells can migrate to the posterior side. So what do you think is happening?
I think that maybe there is, I don't know, I'm elucubrating here, but maybe the axon could be expressing in a gradient these addition molecules. Or maybe there are also like an attractant for these cells. I mean, because in the end the axon provides the scaffold, but maybe the direction is given by the target tissue. Like you have the hair cell here, you have the axon. So neuroblasts will grow on the axon, but they can start doing this maybe. So maybe it is something from the hair cells or the neighboring tissue or the supporting cells that will help us in the end the neuroblasts reach the. So what I think, and I have also seen, is that some of the neuroblasts, they do not just go there and that's it. They go back and forth.
I see. Okay. And then one of the last questions from my side is, so you showed in your work that around 34 hours, everything is pretty much more or less decided, that you have cells which have migrated on the posterior side, and this process starts around, let's say, 24 hours.
The formation of the anterior lobe, I think it starts at 17. When the delamination starts.
So, but what about what you mentioned, that you have these signals from the surrounding tissue which are telling the neuron where not to go. So what is the time on which these tissues arise?
So I don't know if I understood the question.
So okay, let me clarify. What I am saying is that so 17 hour is when you have the first contact on the anterior side, and then more or less there you have a bunch of cells. Then by the time they migrate to the posterior side, already 34 hours, it is 34 hours post fertilization. Now when during this window between 17 to 34 hours, do you have specifications of cells surrounding the aortic vesicle which are secreting signals, which are telling the cell not to extend neurons in that direction? So I'm trying to zoom out from your system and look at the surrounding of where the information could be coming from.
I don't know. I mean, I don't know if this will answer your question, but I think that it could be like several things, like the epithelialization will prevent the cells from ingressing. So once this is finished, it's over. Then the progression of the neurogenic domain from anterior to posterior will give some time specification of these cells that will deploy a molecular toolkit, like timely, no? So the cells that are specified before, maybe they have the capacity to go through cell addition contacts or chemoattracting signal towards the pioneers and not to the rest of the aortic tissue, because it has been, and maybe this will answer better the question, like it has been described that neuro-D will tend to avoid epithelial tissues. So by itself, if you are expressing neuro-D, you want to avoid epithelia. And yeah, once you have that, you go outside. As time follows, you are able to sense the chemoattractant from pioneers. You won't go anywhere else because that is the best place you can go to. Okay. But I don't know if in the rest of the aortic vesicle, I don't know what could be driving like the avoidance of these cells towards, I don't know, instead of, I don't know. Like I think somehow these cells, since the pioneers are here, the delaminating cells are here, it would be really difficult that these cells do like, well, I'm going to the dorsal part of the aortic vesicle by itself because it is easier to go this way. And that is also why the pioneers are there, because maybe if they wouldn't be there, it would be easier for them to go to dorsal parts. Also, I think it's been described that neural crest cells help to the aggregation of this. So maybe if there are neural crest cells surrounding, providing inhibitory signals, so these cells also when they extend philopodia, you can see that at some point they retract and tend to go to other regions. So maybe it's also that. So avoidance of epithelia also reducing the capacity you have to migrate to other tissues from neural crest cells or whatever. And also, in the same line that before we were talking, the CXCL14, maybe since it is if it is an inhibitor of migration, if it is in the hindbrain entry point, so the cells cannot go inside the hindbrain, and so on. I don't know if that answers your question.
No. I mean, my question was quite open-ended, so it's okay.
Okay.
So, and one last question from my side. So I had many sticky notes. I removed them as you were talking. But there is one sentence I would want you to clarify that you wrote in your thesis a bit. So when you in the summary of your chapters 2 and 3, you say that however in our experiment the putative tropic effect of neurotrophins is obscured by its more predominant trophic role. What do you mean?
I mean that if neurotrophins do have a trophic role, it cannot be addressed in our experiments because the trophic role is so much more important that the cells would die. Even before being able, I mean, the axon would be extended, the cell would die, so then, but I mean that any possible trophic role is obscured by its more predominant trophic role, but I still think that they are not, don't have a trophic role, at least for the pioneers axons, because it's been described in mice that BDNF and neurotrophin 3 are important for the outer hair cells or inner hair cells to attach if they express BDNF or neurotrophin 3. So at least in our case for the pioneer axons, I think the only role is trophic.
I see.
But if it had a tropic,
okay,
I don't know.
Okay, thank you.
Thank you.
Thank you very much. So my turn. So I would like to start by congratulating you.
Thanks.
Because, and also Berta, your supervisor, because I think that it's an outstanding thesis. I've been in many already. I'm old, so, and I really enjoy the thesis.
Thank you so much.
I had no idea about the development of the optic vesicle before, so I learned a lot, and the text is so wonderfully written that it was really easy to follow. And actually it was like a good novel, right? Because it was step by step, and you can novel, and you really wanted to know the next step, you know, what was the next step. So it was a really nice text.
Thank you so much.
So I think also for you, I mean, it's not only that it's very nice written, but also technically you have done a lot, and I think that you've been able to, you know, to dissect out different steps of the same process, from you know, early delamination to later astonogenesis. So you have learned a lot, all of the different processes along development. So I think that's been very nice, and. The figures and the movies are beautiful. We have seen it today. It was very, very nice. And I think that you have acquired a very strong background in different things, like you know, cell and molecular biology, genetics, you know, last generation microscopy. And overall, I think that you have integrated, the most important thing that is that you have integrated in your mind the scientific method, which is something that you really need to, that all the students have to learn when they are doing their PhD, right? So congratulations for that, and also Berta, I think you made a very outstanding work. And yeah, most of the questions that I had you clarified during the presentation, or they asked already, but still, I know that you enjoy a lot discussing, so we're going to discuss a bit. Okay, so my first question is how, I mean, maybe it's written, but I didn't see it. So the dominant negative RAG1A, how is done? I mean, how do you know that it's acting as a dominant negative?
Mm-hmm. So
it's published already, or
yeah,
it's published.
Yeah.
So
because in reality, I don't know how it works.
Okay.
But
no, because the dominant negative is always a nightmare. It's not it's not clear how do they.
It's published, because I assume that it might be because the I don't know, the GTP, the domain that makes the GTP go into GDP, it restrains the GTP without a possibility to go into GDP, and if this RAG molecule binds to other RAG molecules, but in reality I have no idea, I'm making it up.
No, actually, that was actually related with my second question, is why didn't you use CRISPR, as you did for to knock down or to knock out this instead?
Because the thing
because it was already published, I guess. No.
So because I mean, you are telling me to use like CRISPR trying to target RAG1.
Yeah.
So the first thing is that we don't have. It would affect the whole tissue.
Right.
But we have a UAS Cas9 that we are setting it up. But in that case, maybe it could be possible to inject the guide RNA only in the UAS Cas9 tissue, which is neurodegal4, would produce that. But the thing is that you have to integrate the guide into the vector, so you cannot inject the ones that you buy, so it's a little bit
much
more
more complicated than I think.
And since it is published, the dominant negatives, I thought that this would provide me with more robust things. Yes. Okay,
good. So there is something that I think I didn't get completely, and it's related a bit with Vika was asking you. So when the axon goes to the posterior pole, I mean, the axon goes to the posterior pole, but then anchor there and then retract, right?
Yeah.
So what happens? So all the neuroblasts go through that are going to integrate the posterior pole, go through that.
Through the axon.
Through the axon?
No.
Or no. So it's just acting as an scout as a pioneer for the first one, right?
Exactly.
Okay.
And the rest maybe also through homotypic interactions if this cell is going. Also
they just follow the pioneers once that they establish the tracks.
I think so. I think so because also in the context where we don't have the axon, the lobe is like scattered. So what provides not only the direction along which the cells will migrate, but also that it is tight enough, is the axon and the cells, the pioneer cells going there.
Okay.
But the rest maybe can crawl on top of the cells going on the axon. Yes.
Right. So that probably is something that is very, I mean, that happens in many other places, right? As you were saying.
In the Drosophila overall, because the pioneer neurons were describing Drosophila, I think a lot of work has been done in invertebrates. They rely a lot on these pioneer neurons. In higher order animals, it's not been so much addressed.
Yeah, but probably because it's difficult to address, because but I'm pretty sure that that something like that happens. In
the end, the idea was not only to study the inner ear, which I made this whole book, but also the cellular processes. Like, do you have these pioneer neurons, and they are so important that they could be shared among other tissues or other ganglions, like neuronal tissues outside the CNS. Also, I got the idea because the radial glial migration, some of the cells can crawl to form the cortex. Like, this should have its homologue outside the CNS, maybe somehow when it turned to be that case.
Okay, another question that I had is, did you try to leave in your when you ablate or eliminate the axon? in all the different cases that you eliminate the axons, did you leave the animals to live longer to see what? No.
No. Yeah, that was a pity because
What is the effect? The real effect of that?
Maybe they have effects on because now Nerea is establishing a protocol for behavioral testing. So maybe, I don't know, if they would have the Crisprs at later stages, the ablation of hair cells at later stages, but no.
Next,
I didn't do anything like that.
the next in the the next person in the lab. Okay. Okay, what else? So why, that's another technical thing, but I don't know much about zebrafish. So why did you start doing experiment with Cas13 and down-regulating instead of doing everything from the beginning with Cas9?
Yeah, that was because people from the lab that were doing the Cas13 experiments had a stronger phenotype. But maybe in their cases was because the proteins that they were targeting were being expressed either maternally or very, very early. So I thought maybe we can get a stronger phenotype with the Cas13 than with the Cas9, or also it could be a proof of concept that the Cas13 is working if it recapitulates the phenotype that we are getting in the Cas9. I think it was mainly these two things, like trying to have a stronger phenotype. In my case, I didn't get a stronger phenotype, but to some extent it recapitulated the Cas9. So, also I like.
If you had to start again, would you do it the same, or?
I think I would stick with the Cas9.
With the Cas9.
I like it more. I don't like to do the mRNA of the Cas13. The Cas9 you can buy the protein.
No, the results are more variable, but it's also because it's RNA, so it's like what else? What good? I don't remember what I. So in your this is interesting. So in your misdirection, axonal direction experiments, when the axons go to the wrong place.
When I overexpress.
When you overexpress it, right? There, what happened with the neuroblast, with the migrating neuroblast? Do they follow that?
The thing is that in those experiments, I cannot. I think I should do more, and I cannot really rely on them, not so much, because I think CXCL14 and neurons or neuroblasts or neuronal tissue does not like CXCL14.
Okay.
So the thing is that I get a lot of cell death.
Okay.
And yeah.
So you cannot see what happened there.
I cannot tell.
But it could be very nice to see.
If it worked, yes. But the thing is that I get so much cell death that, I trusted more the ones that didn't die.
Yeah.
But it was a small proportion from the whole thing. Also in the overexpression in the hindbrain, also a lot of cell death. So it would have been very nice if they hadn't died, because maybe it could have been more robust. But
well, I guess that also you cannot then say, my other question related to that is that it seems that it's not that the normal one don't go to the right place. It's that it seems that there is an extra axon, because in the pictures I see that you still have an axon that go fine, that go to the right place, but then you have like an extra axon that goes that is misdirection, right? So you have like two different.
Like I have.
How do you?
I have quantified it, and normally it's just one axon that fasciculates.
Okay. So it's fasciculation. Okay.
Of this, maybe fasciculation is not the proper term, no, because it would take
the
into account
fasciculation.
several axons.
Yeah.
I don't know, but the thing is that it normally is one, and sometimes if it is done at the middle, you see how it produces a branch. If it is in the region where all the cells are there, maybe you think that there are two cells doing it. Most of the times it is just one cell doing it, but sometimes it could be 2 to 3 also producing the axon.
Okay.
Producing this pioneer axon. So maybe the pioneer is not the pioneer axon, but the pioneer axons sometimes.
Yeah. or branch.
Yeah,
No.
or branches. So maybe in this case, yeah, it is produced the normal one, and then the other one does like this, or it is another cell trying to do it. But as I told you, these experiments, since they have a lot
they
of
are
cells,
difficult.
it would have been so nice because it was so much work trying to clone the.
No, I think it's nice. I think it's still
but
very nice, but yeah.
I
if they
cannot
are
really rely
yeah,
on
okay.
this.
Okay, and so how many molecules did you check before CL14? It was the first one?
It was, I mean, checking with Crispr, it was the first one.
Okay.
In the bibliography, we were looking for
many. So it was the right candidate.
I think I decided that one because actually the real story started.
Yeah, this is what I want. That you tell me the real story.
So this is the story that I told you, and now it's the real story.
Exactly.
So the thing is that we saw CXCL14 very much expressed only in the otic plaque at the time that the pioneers reached there. Okay. So we were trying to find the molecule that made the pioneers go there. Because if we could prevent the pioneer neurons from going there, maybe we could see the effects of the lack of this coalescence region.
Yeah.
It started from that. So, but it was very beautifully expressed in the otic plaque and only in the otic plaque.
So it was so specific.
Exactly.
That
it was
had
like,
to be.
Wow, it could be that.
Yeah. I see. Okay, so and then I wanted to ask you something that I always like to ask the future doctors, and is so from all the different things that are still not answered, but you open and actually you mentioned some of the things that some of the open questions that still not answered. Which one would you like to, if you had resources and the time, which one would you like to follow and to do?
I would go with single-cell RNA-seq of the pioneers comparatively with the delaminated neuroblast to see if their profile is different or just it is a matter of time. So I would go for that, definitely.
Somewhere at the molecular...
because if no, the RNA-seq would provide me with so much information
in
and then
unbiased manner. Yeah.
You can try functional experiments, whatever you want. So this, to have all the information, I would start with that, and then with many more things. Also, I think like the delamination stuff that I didn't go so much into it, like trying to compare if the delamination is an EMT, if they follow, because in the end, no, this tiny tissue has so many things. It delaminates, it migrates, whatever.
No, it's a very nice model. Yeah,
it's a nice model. It's very tiny, but it is nice. So maybe trying to address more into detail this EMT delamination with the neurodigal for line, checking some of the molecules. Also, I'm saying this because you asked me if I had all the resources, and now I came back to I don't have all the resources, but this could be done. But if I had all the resources,
it would be that.
Molecular signature, yes.
Okay, so thank you very much. I think that there is any doctor in the audience that want to ask something? No? The supervisor want to add something on comment? No, no comment.
Aitor has done a great job in setting up many things, and he has been very generous to the lab. So thank you.
That's nice. Okay, so now we're gonna discuss. So please go out, and then we will call you back as soon as we decide something. Okay, welcome back. So, Aitor, after a lot of deliberation, we decided to give you the maximum grade, excellent, cum laude. So, congratulations.
Thank you very much.
That's it. Thank you very much, everybody, for being here. Okay. Yeah. Does anybody want to? He was telling me that I should ask if anybody wants to say something.
No tengo nada preparado. Estoy algo emocionado y nervioso porque, bueno, para mí es una persona muy importante en mi vida. Él lo sabe. Hemos pasado unos ciclos muy complicados a lo largo de nuestra vida. Por más y menos nos ha ido separando la vida. Yo por ceporro, en los estudios. Entonces él iba avanzando y yo me seguía quedando atrás. Pero bueno, hay decisiones muy difíciles que hemos ido tomando a lo largo de la vida, en las cuales una verdadera amistad se demuestra en esos momentos tan difíciles porque uno toma un camino, otro toma otro. Estoy súper contento de seguir estando a tu lado, pese a que la distancia nos haya puesto estos momentos difíciles. Es que estoy muy emocionado, ¿eh? No, ya lloraremos. Y nada, solamente quiero decirte que una persona como tú solamente se merece lo mejor, a pesar de que esta gente te conoce poquito menos de tiempo que yo, que vamos desde la guardería. Solamente te deseo lo mejor, eres para mí lo mejor en esto, porque no hay nadie que te pueda igualar, ya lo has demostrado aquí arriba. Eres una persona inteligente, buena, simpática, atenta, no sé qué más decirte, o sea, me faltan palabras para todo esto. La gente que te ha conocido estos años sabrán cómo eres realmente, no hace falta que tampoco les expliques cómo eres. Y solamente te puedo decir que espero que en el futuro solo te deparen cosas muy buenas, porque ahora mismo tú te la has ganado, tío. Tú te la has ganado, eres una persona que has luchado por esto, has estudiado mucho y pese a las dificultades que hemos pasado durante estos estudios, que tú y yo lo sabemos, has estado, tío, al pie del cañón y aquí lo has demostrado hoy. Simplemente mi mayor enhorabuena, tío. No sé si en boca de todos, supongo que sí, pero por lo menos por mi parte eres una puta máquina. Lo siento por la palabra, pero así lo digo.
Bueno, después de esto, no sé si alguien más quiere decir algo.
Well, apart from thank you, gracias a todo el mundo, sois lo mejor también. Ahora hay un pica-pica en la terraza del PRBB de la tercera planta. Join us in the PRBB third floor terrace from now until you want.
Thank you.
Thank you.